Previously on Ribosome Busters.
And finally, the ribosome.
Oh!
We found a teleporter device and we've managed to go into the ribosome. Let me try it out now.
Okay, let's go.
Oh my gosh.
Go in.
Whoa!
Oh my gosh.
We are finally in the cytoplasm.
Oh wow.
Hey, look, isn't that the ribosome?
Oh, yeah, it is.
It looks like a mitten-shaped organelle with the large subunit and the small subunit. It's 40% proteins and 60% ribosomal RNA.
RNA is a polymer made up of multiple ribonucleotide monomers. Each monomer consists of a ribose sugar, a phosphate group, and a nitrogenous base, all linked together by phosphodiester bonds between the 5' phosphate groups and 3' OH groups of the ribose sugar. This creates a repeating backbone of sugar phosphate units with appendages consisting of the nitrogenous bases. The two free ends of the polymers are distinct. One has a phosphate attached to a 5' carbon, which is the 5' end. The other has a hydroxyl group on a 3' carbon, which is the 3' end.
Proteins are essentially polymers of amino acids. Each AA is linked together by peptide bonds between the amino group and the carboxyl group of amino acids. This creates chains of amino acid residues termed polypeptide chains.
Two free ends of the polymer are also distinct, as in the case of rRNA. One end has a free carboxyl group, and this is known as the C-terminus. The other end has a free amino group, and this is known as the N-terminus. Ribosomes further act as monomer for polymerization, forming a chain of ribosomes, known as polyribosomes or polysomes. During the process of translation, polyribosomes increases the rate of rMNA translation drastically.
Hey, isn't that our lovely AU and buff GC bonds? Nitrogenous bases of the rRNA form complementary base pairs with one another, with adenine forming 2 hydrogen bonds with uracil and guanine forming 3 hydrogen bonds with cytosine.
Apart from rRNA, peptide chains associated within the ribosomes form hydrogen bonds, hydrophobic interactions, disulfide linkages, and ionic bonds within themselves, and with the polar charged rRNA molecules, stabilizing the structure of the ribosome and acting as structural support for the ribosome. Other than intermolecular bonds, there are intramolecular bonds as well. In the large ribosomal subunit, 3 specific binding sites exist for tRNA binding. The A site binds to an amino-acyl tRNA complex, which is a tRNA bound to an amino acid. The P site contains the ester linkage of peptidyl tRNA, which in turn contains the last amino acid of the growing chain. When the amino NH2 group of the amino-acyl tRNA, which also contains amino acid, attacks this ester linkage, a new peptide bond is formed. E site, peptidyl tRNA is moved here after the formation of the peptide bond. The small ribosomal subunit binds to a molecule of mRNA through a binding site. The innate structure of a ribosome is such that it holds both tRNA and mRNA close together, thus making it easier for binding, following the recognition of the codon by the anticodon. This allows the positioning of the new amino acid for addition to the carboxyl end of the growing polypeptide, which in turn catalyzes the formation of the peptide bond. It is important to note that codons and anticodons only bind together when they are of complementary bases. For example, UAG codon would bind to AUC anticodon.
There are 3 steps to synthesize proteins: initiation, elongation, and termination. During initiation, an initiator tRNA carrying methionine binds to the small subunit of the ribosome, bringing together mRNA, a tRNA bearing the first amino acid of the polypeptide, and both the ribosome subunits. The small ribosomal subunit binds to both the mRNA and the initiator tRNA, When binding to the mRNA contains a finder sequence in the 5' end, which binds to a complementary anti-finder sequence of rRNA of ribosome at the mRNA binding site of the ribosome small subunit. The small subunit with initiator tRNA with the anticodon UAC moves downstream along the mRNA until it base pairs with the start codon AUG and the anticodon of the initiator rRNA hydrogen bond to the start codon. The arrival of the large ribosomal subunit completes the initiation process. The methionine initiator tRNA complex is now situated at the P site of the ribosome. Proteins such as eukaryotic translation initiation factor bring all components together, and energy from the hydrolysis of GTP forms the initiation complex. The initiator tRNA will be at the P site, with the A site being available for the tRNA with the next amino acid.
During elongation, amino acids are added one by one to the first amino acid. Each addition involves several proteins called elongation factors and occurs in a 3-step cycle. The first step is called codon recognition. The mRNA codon in the A site of the ribosome forms hydrogen bonds with the anticodon of an incoming molecule of tRNA carrying an amino acid. An elongation factor, which is one of the proteins, ushers the tRNA into the A site. This step requires 2 molecules of GTP, with the P site still being occupied by the initiator tRNA complex. The second step is called the peptide bond formation. A rRNA molecule from the large ribosomal subunit, acting as a ribozyme, catalyzes the formation of a peptide bond that joins the carboxyl end of the polypeptide, extending from the P site to the newly arrived amino acid in the A site. This step removes the polypeptide from the tRNA in the P site and attaches it to the amino acid on the tRNA in the A site, bringing together the 2 amino acids. tRNA at P site loses its amino acid in the process catalyzed by peptidyl transferase, part of the large 60S ribosomal subunit. The last step of the elongation stage is called translocation. The tRNA that was in the P site is moved to the E site, where it leaves the ribosome. The A site now contains the next codon to be read, where a new tRNA carrying the next amino acid enters. At the same time, the ribosome moves the tRNA with its attached polypeptide in the A site to the P site. As its anticodon remains hydrogen bonded to the mRNA codon, the mRNA moves along with it down 3 nucleotides in a 5' to 3' direction, and brings the next codon to be translated into the A site. This step requires energy, which is provided by 1 molecule of GTP. The ribosome and the mRNA move together in one direction, codon by codon. The elongation cycle is rapid and repeats with every amino acid added to the chain till the polypeptide is completed.
The final stage is termination. Elongation stops when the ribosome reaches a stop codon, UAG, UGA, or UAA as shown in our mRNA. These acts as stop signals. At the A site, a protein called the release factor binds directly to the stop codon. The release factor causes a water molecule to be added to the polypeptide chain in place of an amino acid, resulting in the hydrolyzation of the completed polypeptide from the tRNA at the P site, thus freeing the polypeptide through the tunnel of the ribosome large subunit. The 2 ribosomal subunits and other components of the assembly dissociate.
Oh my god, look at our polypeptide.
Yeah, it's so awesome, but I'm hungry. Let's get out of this ribosome. Oh no no. Ribose sugar. Okay, can you line it according to ribose sugar?
Why did we put it on the board?